Welcome everyone. My name is Garth Herring, an avian ecologist with the U.S. Geological Survey Forest and Rangeland Ecosystems Science Center in Corvallis, Oregon. Today, with my co-principal investigator, Colin Eaglesmith, we will be discussing why understanding prey availability in terminal lakes is important to waterbirds, our approaches used, and some general results from this first full season of research. The title of this presentation is Great Basin Terminal Lakes Waterbird Prey Availability. Just a quick reminder of the origins of this USGS study. These studies were developed after the Saline Lakes Ecosystems in the Great Basin States Program Act of 2022 was signed. The overarching objective of the Saline Lakes Integrated Water Availability Assessment Project is to improve understanding of the impacts of water management decisions on the migratory waterbirds of the Saline Lake ecosystems in the Great Basin. The overall project is organized around 5 science pillars: waterbird movement, waterbird aquatic prey, food, resources, and habitats, also referred to as waterbird prey availability, water quality, water quantity, and remote sensing. Note that the 5th science pillar, remote sensing, was added later to support the other science pillars such as waterbird movements and prey availability, and generate additional critical data such as lake bathymetry and lake spatial extents. However, you might be wondering how we settled on the initial 4 science priorities. Before we talk about what research we're doing across the Great Basin to understand waterbird prey availability, we thought it would be beneficial to provide some background on the process used to arrive at these priorities. When the Saline Lakes Ecosystems in the Great Basin States Program Act of 2022 was signed and the USGS team was first put together, early conversations recognized the importance of identifying existing research and understanding stakeholder perspectives. To address this, we initiated a literature synthesis and gap analysis and conducted stakeholder engagements to inform our understanding of terminal lakes science needs. To initially identify previously published research papers, reports, theses, and dissertations, we used a keyword search for terminal lakes ecological and hydrological terms using both Google Scholar and Scopus. Our initial queries identified over 9,000 documents related to the ecology and hydrology of terminal lakes. We then utilized a large team of USGS ecologists and hydrologists to rapidly review those 9,000 plus documents, basically titles and abstracts only, to determine relevancy to Great Basin terminal lakes. That review yielded nearly 800 documents. The remaining 8,200 documents were largely associated with terminal lakes around the world or were only tangentially related to terminal lakes. Those final documents were reviewed and assigned to broad ecological and hydrological categories, and we identified what lakes were associated with. We then conducted an information density analysis by lakes to identify gaps in our understanding of terminal lakes in the Great Basin. Without getting into the results, the primary takeaway from this synthesis is that for a few lakes we have a lot of information, and for most lakes we have little information. To obtain stakeholder feedback on ecological and hydrological research priorities, we held 3 separate workshops for lakes in California and Nevada, Oregon, and Utah, with over 120 participants attending. We utilized polling software to help prioritize ecological and hydrological information needs. We also asked participants to rank the differing waterbird groups from the perspective of conservation and future research needs. Based on stakeholder feedback, ecological priorities included studies on habitat, waterbird dispersal or movements, waterbird population abundance, food or prey availability, and threatened and endangered species. Hydrological priorities based on stakeholder feedback included water budgets, water use, lake or wetland surface area, hydrologic vulnerability, and water quality. When coupled with our literature synthesis, these priorities are what ultimately define our 4 science pillars: waterbird movement, waterbird prey availability, water quantity, and water quality. That stakeholder feedback was overlaid with the literature synthesis results to ultimately define the science pillars. Lastly, our stakeholder engagement polling informed our prioritization of waterbird guilds, with shorebirds being the highest priority based on long-term population declines and critical habitats provided by terminal lakes during shorebird migration. Now we're going to pivot from how we defined what research was needed across the terminal lakes to our actual research focus on prey availability. Note that prey availability is critical to most aspects of the annual water, including movements and habitat selection, reproduction, physiology, and survival. When we looked at the Great Basin to identify lakes for sampling, we largely focused on those lakes that were identified as important lakes for shorebird migration from the literature. Those lakes include Malheur, Abert, and Summer in Oregon, Honey and Mono in California, Carson Sink, Stillwater, and Carson Lake in Nevada, and Great Salt Lake in Utah. These lakes also represent a salinity gradient from freshwater to hypersaline, with replicates within each salinity category. Our shorebird prey sampling utilizes a repeated sampling framework with sampling depths specified by shorebird foraging depths. For shorebirds like phalaropes that are not tied to shallow waters, we are sampling at deep water sites. Note that this also allows us to understand prey availability for important open water birds like eared grebes, which the terminal lakes support over 90% of the North American population at some point in their annual cycle at Great Salt Lake, Lake Abert, and Mono Lake. Finally, we are collecting water chemistry data at invertebrate sampling sites to understand responses to changing water chemistry across space and time. Prey availability sampling allows us to assess community composition, density, and biomass of prey, the caloric value of prey, and how these change across space and time, and in association with changing water chemistry. While some of these details may be known at some lakes, they generally are not well understood across the entire system. Additionally, changes to the caloric value of these invertebrates throughout the year as water chemistry, particularly salinity, is not well understood, but typically increases in salinity approaching critical thresholds result in declining invertebrate quality. For species like shorebirds, whose long-distance flights depend on these lakes for migratory fueling, declining prey quality or energetic value could have important ramifications. The overall goal of this research is to utilize these estimates of prey energetics, habitat availability from remote sensing research, and shorebird energetic needs to model lake shorebird carrying capacity estimates, or how many birds can a lake support and for how long. Those estimates could then be used to evaluate how changes in lake volumes or extent and water quality would influence prey availability, with consequences for shorebird carrying capacity and potentially waterbird movements both within and across lakes. To address these research needs in 2024, we sampled invertebrates across these lakes from April to October. Across the 9 lakes, we sampled over 2,200 individual samples at over 390 sites. Sample processing in the laboratory is ongoing, and we expect to have completed processing before initiating the 2025 season sampling in March of 2025. As an example of our sampling during 2024, we can look at Lake Abert. Although we're using a repeated sampling approach, that is sampling at the same site monthly, those sites tend to move each month as water levels recede across the season. Subsequently, the shallow water or shoreline sites continue to extend further out into the lake. This is necessary to continue to sample the appropriate depths for shorebirds across time. Also note that portions of Lake Abert have no sampling effort because they were dry during this season. Looking ahead to the 2025 field season, we plan on refining some of our sampling methods to better capture variability in certain invertebrate groups like brine and fly larvae. We would also like to add some additional lakes, such as Eagle Lake, to better understand how these lesser important lakes, from a shorebird perspective, could potentially support more birds in years when other lakes in the system might be dry. Lastly, we'd like to add a limited amount of sampling at sites where waterbirds are actively foraging across the lakes to better understand habitat selection processes. We would like to acknowledge the USGS Water and Ecosystem Mission Areas for supporting this research, our fellow USGS Science Pillar researchers, and collaborators at the U.S. Fish and Wildlife Service, Utah State University, Utah Department of Natural Resources, UC Santa Barbara, and Westminster University. Lastly, our staff, notably Harris Kopp and Julie Prang, who have worked tirelessly throughout this long field season. We appreciate your time to listen to this presentation on the prey availability science pillar. This concludes this presentation. I will take questions if there are any. Thank you.